Hello everyone. Today I'm excited to present our work, Nearest Neighbor CCP-based molecular sequence analysis, an efficient approach we have developed for handling high-dimensional biological data. I am Sarwan, and this is a joint work with my colleagues from Georgia State and Georgia Tech. So the core problem we are trying to address today is how to make molecular sequence analysis scalable while maintaining the accuracy. So let me talk you through what we'll cover today. So we'll start with the problem formulation and motivation, essentially why existing methods aren't uh, cutting it for the large-scale biological data. Then I'll give you a brief overview of the correlated clustering and projected method, or CCP, which forms the foundation for our approach. The main contribution is our proposed CCP-NN, or the nearest neighbor algorithm, where we'll dive deeper into the technical details. We'll then analyze the complexity improvement we achieve, provide theoretical convergence guarantees, and validate our approach on real-world biological data sets. Finally, I'll discuss the technical insights and the future direction this work opens up. So let's start with the fundamental challenge. When we are dealing with the molecular sequences, whether they are proteins, DNA, or RNA, we are essentially working in extremely high dimensional space. The curse of dimensionality hit us hard here. So we have feature space in R to the M, where M is the number of features, is often much larger than N, or number of sample, it's much larger than N, which is our number of samples. This creates sparse representation that are computationally expensive to work with. The real killer is the computational complexity. So most existing methods require n squared times m pairwise computations, and they need to store in O to the n square distance matrix in memory. When this n gets larger, say tens of thousands of sequences, this becomes completely intractable. Now, existing dimensional reduction methods have their own limitations. So linear methods like PCA assume linear relationships which rarely hold in the biological data. So what about the nonlinear methods? So they are computationally prohibitive for larger data sets. So CCP shows promise, but it still has this n square bottleneck. So you can see the scalability problem clear in this figure in this slide. So as n increase, we get this quadratic growth in runtime, while what we really need is something closer to the linear scaling. So let me briefly explain the CCP algorithm that form our foundation. So CCP take the data sets X in R to the n times M, which finds the low-dimensional embedding that preserve important structure relationships. The CCP pipeline has several key steps. First, we do the variance-based feature selection. We keep only the top features which actually vary across our samples. Then we use the k-mean to partition these features into clusters. The critical step is the density map computation. For each cluster C sub I containing feature f of I, so we compute a density function rho I, which is where the magic happens. So we are essentially measuring how correlated the features within each cluster are. So if you see this equation, so that then the density computation uses the kernel function k, either exponential or Lorentz, applied to the pairwise distance between features. The scale parameter controls how local or global these correlations are. So this gives us a meaningful low dimensional representation that captures the underlying structure of our biological sequences. Now let's look into at the algorithmic details of how CCP compute these correlations. So this is important because it's exactly where our bottleneck occurs. So the algorithm compute pairwise distances either between all reference samples if we are doing a transform or within the dataset itself. So the key insight here is in line 7 through 15. So we compute the average minimum distance to set or scale parameter adaptively. So we also set a cutoff based on the mean plus 3 standard devs of the distance distribution. This helps us focus on the meaningful correlations and ignore the noise. But here's a problem. That pairwise distance computation in line 3 or 5. So that's the n-square operation. And it's computational bottleneck that limits the scalability. Now, um, so this brings us to our key innovation, which is the CCP nearest neighbor algorithm. So the core idea is beautifully simple. Instead of computing the exact pairwise distance, we use the approximate nearest neighbor search. So we use the NOI library, which builds a forest of random projection trees. The beauty of this data structure is in its complexity profile. Buil Building the index takes O to the n log n times f times overall runtime, but each query is only O to the log n time, right? And the space complexity O to the space complexity is big O of n times f, which is much more manageable than the n square. The approximation quality is controlled, so we can bound the expected error in our nearest neighbor approximation. So this is crucial because it means we can provide theoretical guarantees about our method's performance. Each tree in the forest uses random projection to partition the space. So when we query for the nearest neighbor, we traverse multiple trees and aggregate the results. The elegant part of that we only modify step 5 of the original CCP algorithm, which is the density computation. Everything else remains identical. So we preserve the fundamental approach while gaining the massive computational saving. So let's dive into the technical implementation. The nearest neighbor compute correlation function replaces uh, it replaces the exact distance computation with approximate nearest neighbor search. First, we build an NOI index with the appropriate metric. This could be Euclidean, cosine, or other distance measured depending on the data. So we add all our reference vectors to the index and build it with all available trees for maximum accuracy. The key difference is in line 8 and 10. If you see here and here. So instead of computing all pairwise distances, we query for the k nearest neighbor for each point. This transforms our n square operation into n log n. Importantly, we keep the same scaling and cutoff computation as in the original CCP. This ensures that our density estimation remain meaningful and comparable to the exact method. Now let's analyze the theoretical complexity improvement. This is where we can really see the impact of our approach. So the original CCP algorithm has a time complexity of n times f to the n c times the total number of iterations plus n, which is dominated by the n square times f term for pairwise distance computation. The space complexity is n times m plus n due to those distance matrices. For our nearest neighbor, CCP-NN, it reduces this to n times f, and again nc times the number of iterations plus log of n, which is dominated by the n log n times f. The space complexity become n times m plus log n times f. The asymptotic improvement give us a speed of factor of n over n log or over log n. And for concrete number, if we have 10,000 samples, 100 features, and 10 clusters, so the CCP will require about 1 billion operations, while the CCP-NN need only 1.3 million operations. So this is a theoretical speed up of roughly 770 times. And this improvement only get better as an increase. So of course, we need to ensure that our approximation doesn't hurt the quality of our results. So let me walk you through the theoretical convergence guarantees. So we assume our data X is drawn from some distribution P of X with the density P of like the small P of X. The NI index provide the epsilon approximation nearest neighbor. We can bound the expected error in our neighbor selection. Or CCP-NN density estimate is computed using these approximate neighbors. The key insight is that we can bound the error in our density estimation using the triangle inequality. The final result shows that our expected error is bounded by three terms: the approximation error epsilon, a bias term H4, and a variance term 1 over KH, these ones. So this tells us that we can control the approximation quality by adjusting our parameters. Importantly, this bound decrease as we increase K, so the number, which is the number of nearest neighbors, and it's directly related to the quality of our approximation index. So let me discuss our experimental validation. So we tested our method on the three diverse biological data sets to ensure our method work across different type of molecular sequences. So we use a protein subcellular data set that has around nearly 6,000 protein sequences across 11 subcellular locations. The coronavirus host dataset contain 550 around over 5,500 spike protein sequences from 21 different host species, and the human DNA dataset has 4,380 nucleotide sequences across seven gene families. We tested four different embedding methods, from simple one-hot encoding to sophisticated neural network embedding. So this let us see how our method perform across different feature representations. Our evaluation was comprehensive. Seven different classifiers, 5-fold cross-validation, repeated 5 times. So that's 175 experiments per method, giving us a robust statistical validation. So now for the exciting part or results, let me highlight the key findings for the protein subcellular data. First, the accuracy. So across all embedding methods, the CCP-NN consistently outperformed the original CCP, which with one hot encoding, we see a 5.6% improvement. With the spike to VEC, it's a 3.6. For PWM to VEC, it's 4.1. And with the autoencoder, it's 4.4% improvement. But the runtime results are even more impressive. So with the one hot encoding, we get 1.3 times speed up, modest but significant. With spike to VEC, we achieve 3.6% speed up. With PWM to VEC, it's 2.3% improvement. But here is the kicker. With autoencoder embedding, we achieve 14 times speed up. So this shows that our method scale particularly well with the dense high quality embedding. The key insight is that better approximation quality. The key insight is that the better approximation quality, which comes from using our informative embeddings, actually lead to improved classification performance, not just the faster computation. So let's look at the scalability in practice. So this graph here in this slide shows the runtime versus the dataset size for both methods, the original CCP and the nearest neighbor based CCP, which we are proposing. So you can clearly see the quadratic growth pattern for the CCP as the dataset size increase. The runtime grows rapidly. By contrast, the CCP-NN exhibits near linear scaling behavior. The memory usage tells a similar story. So CCP require n square storage for distance matrix, while CCP-NN only needs n log n for the index structure. The practical impact is clear. For dataset with more than 10,000 samples, CCP become computationally prohibitive, while CCP-NN remain attractable even for the larger datasets. So let me summarize our key technical contributions. From an algorithmic perspective, we have created a hybrid approach that combines CCP's correlation-based clustering with the efficiency of the approximate nearest neighbor search. We provide theoretical guarantees of formal convergence analysis with the bound approximation error. So this isn't just an engineering optimization, it's a theoretically grounded improvement. The practical scalability speaks for itself. So we have reduced the complexity from n squared to n log n, making large-scale analysis feasible. For empirical validation shows accuracy improvement of 10.8% and the runtime speed up of 14 times on the real biological datasets. Importantly, these improvements are robust across multiple embedding methods. For future work, we are exploring deep integration with the transformer-based protein language models, multi-scale analysis for hierarchical biological structures, and distributed computing improvement for genomic scale datasets. Okay, so let me just conclude by emphasizing that the CCP-NN what it achieves, it bridges the theoretical and the practice in computational biology. For theoretical contribution include rigor, complexity analysis, convergence guarantee, and bounded approximation error. So these aren't just the mathematical exercise, they provide confidence in our method's reliability. The practical impact is transformative. We enable large-scale sequence analysis that was previously computationally intractable. So we maintain and often improve classification quality while achieving significant computational saving. The CCP-NN delivers on four key promises: better accuracy, fast runtime, lower memory usage, and true scalability. This opens up new possibilities for analyzing large-scale biological datasets that were previously out of reach. Okay, thank you for your attention. I would be happy to take any questions and discuss any aspects of our work in more details. You can reach out to me over my email if you would like to discuss more or collaborate or have further questions. Thank you.